ask the next speaker, Krystian Komenda. I do not have the information from which scientific faculty you are. Maybe you will tell us this during the presentation. And the title of presentation is Simulation of the Molecular Dynamics of the System containing hydroxyectoine molecules covering the cell membrane of red algae, Cyanidioschyzon merolae. Oh my God, you want to kill me with these names. Krystian, the space is yours, and maybe you will tell us how to pronounce correctly this algae.
Okay, at first I need to ask you about, can you hear me and see?
Yes, of course.
Everything is fine. Okay, so as you said, as you mentioned at the start, the main field of study is on the verge of bioinformatics, is molecular dynamics simulation system. So it's not just a particular bioinformatics.
Krystian, one thing, do you want to share your screen with the presentation?
Yes, I'm still working on that.
Okay.
Give me 1 minute. I will read out.
Okay, perfect.
Kind of share screen.
Okay, now it's visible, but just show us only the presentation, because now we see all the slides.
Okay, yeah. I hope that everything will be fine. Okay, so the main topic I decided to present on this conference is simulation of molecular dynamics of lipid membrane. Is kind of a shortcut of that example I presented. What is that, and what type of study you need to go into to understand and to get more information? So molecular dynamics is kind of field between computational chemistry and bioinformatics, and it's widely used to simulate interactions between membranes, between lipids, between micromolecular scales inside what is going on the processes inside the cell or interacting with the cell. So the topic of my presentation is simulation of exactly molecular dynamics of the system who contains hydroxyectoine and ectoine molecules that are covering the cell membrane of red algae. This red algae is, the full name is totally extremely hard to pronounce. However, probably we should use the word Cyanidioschyzon merolae. Of course, it's the only Latin name. We don't have specific information. We don't have Polish and English name for that micro red algae. So, what is Cyanidioschyzon merolae? At first, we need to, because we need to understand how the system works and how to use molecular dynamics. Cyanidioschyzon merolae is a small single eukaryotic cells of from the part of Rhodophyta. It lives in external thermal water environment with temperatures extremely high, up to 45 degrees. Also, it can this microalgae can live and survive in small lakes close to volcanoes. So that's extremely harsh environment for the organism, especially for the microalgae. However, this microalgae can cope with the huge temperature, with acidity, because it can survive in extremely low pH, close to 1.5. And what is important to that microalgae? It's really small. It has only 2 micrometer diameter, and it doesn't have any cell wall. So with that information, is the microalgae has a lot of capabilities to sustain in such harsh and uninhabited place like thermal waters. So that is a perfect model organism for space research, and it has huge adaptation. It can adaptate with UV radiation and osmotic stress. So this microalgae could be a potential to introduce this microorganism in space production and space industry. So of course, because we know how microalgae works, they can produce a lot of oxygen, reducing of course at the same time CO2, carbon dioxide. So this can be used in life support systems that can help astronauts to sustain in such unhealthy places and habitats on the space orbit, or of course on the habitats on the Moon or future on the Mars. Okay, so moving on, what is ectoine and hydroxyectoine? Those substances are derived from ketones, and also it works. They are naturally produced by some kind of extremophilic bacteria. They are producing ectoine, both and hydroxyectoine, to protect the bacteria from osmotic stress, from UV radiation, and from dryness. How it's working? Because of the compound and bipolar electricity on the edges of the ectoine and hydroxyectoine, it can aggregate together and accumulate and interact highly with the water. So it can interact with the water, bind with them, and then create some kind of medium aggregate just to protect from radiation. So my task was to prepare at first I need to ask a question. Are merolae good model for organism in space research? Then I needed to examine the interaction between substances in the simulation box, in the simulation system hall, when I prepared that. And after all, the last chapter of my work was the simulation was the exact simulation of molecular dynamics of this lipid, of the lipid membrane that were, interacting with ectoine and 5-hydroxyectoine in that particular red microalgae. So how I prepared that? I prepared firstly membrane bilayer that consisted MGDG, that's a monogalactyso diacylglycerol, and that's a main compound of the cell membrane that consists in red algae, the particular merolae. Then I introduced ectoine and both hydroxyectoine in solution 1%, 2%, and 5%, and I put the simulation for 100 nanoseconds. Then I measured density profile, membrane thickness, and also number of hydrogen bonds. That could help me to understand how can ectoine and hydroxyectoine interact with the water and interact with the membrane and also interact with each other. Both, not because I'm talking about ectoine and hydroxyectoine. I didn't mix the ectoine and hydroxyectoine, that wouldn't work. So the results were presented on several graphs and gave me kind of results which helped me to understand how ectoine and hydroxyectoine could work and how it's really working. So at first of my results were that ectoine and hydroxyectoine, both, they do not intercalate in lipid membrane. They just stay at the surface or just lay in the layer closely related to the membrane. So I didn't notice a lot of interactions between a particular membrane and lipid composition close related to the cell membrane. However, I noticed by the hydrogen bonds and density profile that the most interactions are with the water. It was approximately 10 times more interactions between water and the cell membrane. And also regarding to density profile, I noticed that both ectoine and hydroxyectoine can aggregate in order to protect because they were aggregating in some parts in some field closely related to cell membrane. And that was a conclusion that could aggregate with close to the membrane in order to protect them, protect the lipid membrane. And to summarize everything what I said, because it was a kind of short preview presentation of everything. At first, I need to tell that hydroxyectoine works as fine as ectoine in contacting with the water. If interacting with the water, I saw that hydroxyectoine has, because of the composition, it has one OH group on the in the composition. It can also interact more with the water instead of ectoine, and it has more abilities to aggregate because of the all particular charge. So to sum up everything, I need to tell that hydroxyectoine could be a really good example of helping micro red algae, merolae, to protect it from UV and osmotic stress, because just of the water binding and preventing cell membrane or whole microalgae from drying and from the problems that could be caused in the low orbit, in orbit stations, or in the future places like in a habitat on the moon or on the Mars. So this is a small step to prepare and to understand how this could work. And that's one answer that molecular dynamics and bioinformatics can help to explore and expand the knowledge in space field and space science and space industry. Thank you for my attention. And if you have any questions, I will be glad to hear it.
Okay. Guys, dear all, we have a lot of time for the questions. We have the 7 minutes, and if you have any questions, please write them on the chat. I see the question Arkadiusz has: are there any plans to researches on merolae in other space or in reduced gravity conditions, for example, parabolic flights?
Merolae is kind of is really extremely simple organism that live on Earth, but we still don't have much information how is exactly lipid composition inside. However, my focus and my view was kind of new in the whole study and whole industry, because nobody ever before discovered and probably tried to use merolae in space industry. So that's kind of new topic, a new approach. However, I think that if we could manage this project, and because regarding to the high capabilities of survival, we can, in the future, there will be more experiments in space, outer space, or reduced gravity. However, we can work with merolae with reduced gravity. Also on Earth, it's not only about parabolic flight. We can use random positioning machines that they are reducing. They have special spin motor that can reduce the gravitational pull force. So we can work also with that on merolae in the future. And it couldn't be really extremely cost because of the RPM machine that is much cheaper than preparing a parabolic flight or even sending merolae to space.
Okay, thank you. We have next questions. Which merolae strain you are using? 10D?
Okay, so I didn't approach for the special strain of merolae because I used only molecular dynamics and on simulation. So that's a kind of, I only needed to know how its exact lipid composition, and that was probably from the, I'm not sure about the strain that was used. However, the origins and the information about the data showed me exact lipid composition, and I was obligated to use it on my simulation. However, regarding to construction and the problem of computational problem and the time of calculating on the computer, I needed to take approximation. So using 10D strain or another type of strain, it doesn't really matter in molecular dynamics simulations.
Okay, thank you. And the next question, can you please tell me about the environment it was isolated from? Perhaps I missed it. This is the question from Priya. The last one was from Julia, and the next one is from Priya. Sorry.
Mm-hmm. Okay, the place where they first isolated from, probably you may ask about it because okay, so it firstly was on one of the most important places where isolated was in probably in United States, in hot springs. So that where the merolae were discovered. However, as I mentioned before, I didn't work because of the it's a kind of simulation, so I didn't work on exactly part of microalgae I used before. I work in a lab that was just took from the basic data, just what I took from the preview publications. That way they were working on the microalgae, probably isolated. One of them was isolated in hot springs in Colorado. So that was the data I took from that. However, it also depends on the place on Earth where can be isolated, where can be took as a sample organism.
Okay, we don't have any other questions that we can say. Just thank you, Krystian, for your presentation, and we can start for the next one.